What we're looking at here is an observable notebook, which we have developed to present a condensed, high-level visualization of large collections of intrahost variants computed from multiple deep sequencing data sets, Illumina or Oxford Nanopore, using Galaxy workflows. Before I describe what Observable is, let me just show you briefly what an observable notebook looks like. So what I'm showing you here is an interactive notebook. I will demonstrate its features in a moment, that presents summary and detailed data about thousands of individual deep sequencing data sets from SARS-CoV-2. In this particular case, what I'm showing you is a data set of about 1,700 samples collected from Estonia, collected over this time period from early fall of 2020 to early April of 2021. Here you can see some basic description of what kind of variants you can see in this data set. So about 1,100 unique allelic variants. I'll show you again what each one of those looks like. There's about 33 of those differences per sample, meaning on average a sample differs from the reference SARS-CoV-2 genome at 33 positions. The vast majority of them, about 30, are fixed, which means they occur at very high frequency, so say over 80%. And there are about 1.5 that are low frequency, which means that occur in, say, fewer than 25% of the mapped reads. So the interactive feature of this notebook, which I will show you right now, is that, for instance, I want to only look at more recent samples from March to April. As I click this recompute button, everything in this notebook display below updated. Let me show you this to you again. And in fact, every time I make a change like this, I select something from a widget that allows user interaction, either automatically or following a button click, the notebook updates. So here's another example for you, just sliding around. No particular reason to do this. I just want to demonstrate the ability to do so. So let me go back and clear the selection, recompute everything again, and show you a few more things down the line. So what you see here is a different view of the same data. Instead of showing you summaries, I'm digging a little deeper into spatial distribution of variants. So the plot below shows normalized allelic variant density, with allelic variants stratified by type, indel, non-synonymous, stop, and synonymous. And what you see on the x-axis are individual SARS-CoV-2 genes and open reading frames. The size of the bubble shows you a plot density. So, the bigger the bubble, the more variants you see. For example, you see that the vast majority of stop variants occur in ORF8. And if you look at non-synonymous variants, there's sort of a lot of genes that have very few of them, say, the matrix gene, and things like spike have a fair number. So you can see there's a pop over there, and the color indicates, when you do find these variants, what frequency with the sample are they at? Are they fixed or are they low frequency? For instance, if I wanted to only look at low frequency variants, I could slide this. Notice everything in the notebook updates immediately and automatically. The plot, the text, and even below you can see the list of individual variants that occur between, say, 5% and, in this case, 15.8% allelic frequency. If you have metadata about samples, you can ask questions about which lineages of SARS-CoV-2 are prevalent. So this is a busy plot. I can actually make it less busy by just looking at monthly aggregation. You can see that starting in February of 2021, the predominant lineage in Estonia, not too surprisingly, was the alpha lineage, or B117, although there's quite a bit of distribution of other lineages as well. And if I want to look at the occurrence of specific mutations, I can do that as well. So, I can just go to this box, and for example, I want to look at N501Y, which is the canonical variant for alpha, beta, and gamma lineages. And you can see that in Estonia, this variant occurred in 914 of 1,691 samples. the most of the time this variant occurs at very high frequencies within the sample, so over 80%, meaning it's fixed. There are a couple of samples here where it occurs at low frequency. So let me go back to the top and load a much larger data set just to indicate that this notebook is capable of analyzing a fairly large volumes of data directly within the browser. I would like to emphasize that all the processing occurs in the browser, in the JavaScript engine. There is the only thing that servers are providing are flat data files, in this case compressed JSON files. So in this case, we're looking at 140,519 samples, which is about 100 times more than last time. And it took a couple of seconds to render, but nonetheless, the notebook is capable of doing it. You have already seen this plot, a very different distribution of lineages because it's now later. It goes past April of 2021, so you can see this COG-UK data set, which is from the United Kingdom. In the last 2 months, there are most of the samples that we collect are assigned to B167.2, which is the Delta lineage. Now that we've seen a little bit of what this notebook is, let me show you just a few behind-the-scenes components. So the first thing that you will notice is that the code for everything that we need to render and process this dashboard is present in the document. So it's basically a self-documenting notebook for those familiar with things like Jupyter. This is analogous in some ways. So all of the, for example, we can look at the bottom, you can see we import things like the D3 library. We import common JavaScript library Lodash, a plotting library called VegaLite. We import modules from other notebooks, ours, and from other users. That's one of the things that is very powerful for Observable. It's very easy to reuse snippets and libraries, either from standard distributions and from other notebooks. You can see, data arrays and so on. If you, for example, want to see how these plots are generated, these plots are all generated with VegaLite, which is a very popular plotting library based on top of D3. It's a declarative graphical grammar. You can definitely modify it, review it, and add new plots as you like. So here's, for example, a view of where the notebook is pulling its data from. So we can see that here you have a widget that it shows you which countries we have processed, options, so COG-UK, Estonia, Greece, and Ireland. And down here, we have corresponding URLs, which just pull data straight from GitHub. So from our GitHub repository, which you can access and view, we regularly update it. And obviously, if you have your own way to distribute data like this, you will be able to, pull it into an observable notebook and view it. Next, I'm going to quickly demonstrate how it is possible to import data that are the end result of a Galaxy workflow, something like this, which is a tab-separated file containing variant calls from many different samples, and import that into an observable notebook. I'm going to retrieve a Galaxy summary file to a local system. Display it, make sure that's what it is, then compress it. All I'm going to do is I'm going to place it in our GitHub repository. So we're going to do git add variants, commit it, and push it. Enter host variants, example. I'm going to next import these data into a different observable notebook, which is also something we've developed for variant file exploration. So all I'm going to do here is utilize the feature of the notebook which accepts arguments. So URL argument, which I've simply now pasted a direct download link for the variance file we just uploaded to GitHub. And I'm further going to indicate that this is a TSV file. So now I have this data set loaded. There are 88 samples. There's nothing selected. What I can do now is, scroll through this very long list of options, which indicates all the individual variants that are present in these files. I can select one. For example, this variant occurring in position 28881 is a G2A mutation that is found in just over a third of the samples. It is a variant, when it's found, it's fixed. It's a non-synonymous variant, and it's a substitution from an R to a K at position 203 of nucleocapsid protein. Here's the distribution of its allele frequencies. And here I have some other interesting information, which basically tells me how does this variant co-occur with others. So this is a simple linkage disequilibrium or co-occurrence test, which says that this variant with the selected 2881 GA co-occurs effectively perfectly with the other two variants, 2882 G2A and 2883 G2C. So their linkage disequilibrium is effectively 1. And you will see that these variants, the red variant is the variant of interest, and these two variants co-occur with it in effectively every sample. So this is a well-known triplet mutation in the nucleoprotein region of SARS-CoV-2, which almost always co-occur in these triplets. Finally, I'm going to demonstrate very quickly how we could make small modifications to this notebook and immediately have them reflected in the code and the display. So for instance, here at the moment, the way these variants are listed, they only show us the nucleotide coordinate and the nucleotides being changed. Say I want to also add to this, for easier access, the name of the protein and the amino acid change. So first, let's see this notebook element pulls its data from a variable called bivariant. So I'm going to scroll down and identify where this bivariant exists. So here is bivariant. This bivariant itself is generated from a variant table object, which is here. This is the raw data for all the variants, and you can see that the variants themselves are now just JSON-like objects, JavaScript objects, with all the attributes that were TSV file are present here. So for example, we can also add amino acid and the name of the gene that's being used. So here, bivariant uses a function called variant key, which creates something that is going to index this variant. So let's look up the function called variant key. It is here. And all it does, it takes a row of attributes for each of the variants and creates a string identifier for it. So let's just modify it. Add a space, add the identification of the gene, another space, and identification for the amino acid. So we've made this change. Now if you go up here, you will notice that all the variants have been updated. So for example, we can now look for variant by amino acid. So L452R, which is a diagnostic variant for the delta lineage. And we can see that it's now found in 12 out of 88 mutations. One final thing that I would like to draw your attention to is this notebook has now been modified, and it is not visible to other users of Observable until I publish it. So this is very similar to creating a branch in Git for your code, modifying it, and then pushing it back. So if I'm happy with these changes, I hit republish, and now everything that we've just done is visible to the public.